Amino acid catabolism involves three steps: removal of amino group from amino acids, that is, amino acid deamination, and conversion of amino groups to ammonia, then the formation of urea from this ammonia in urea cycle, and the conversion of amino acids carbon skeletons into metabolic intermediates like TCA intermediates or their precursors so that they can be metabolized to carbon dioxide and water or used in gluconeogenesis. Amino acid deamination involves two types of biochemical reactions: transamination and oxidative deamination. Transamination is the dominant reaction in removing amino acid nitrogen. It involves moving an amino group from a donor amino acid to the keto carbon of an acceptor alpha keto acid. As a result of this transfer, alpha keto derivative of the amino acid and corresponding amino acid is formed. All amino acids except lysine, threonine, and proline participate in transamination reactions during catabolism. It is a readily reversible reaction and is catalyzed by the enzyme aminotransferases. The enzyme aminotransferases are named after the specific amino group donor, and this is so because the acceptor of the amino group is almost always alpha keto glutarate. By transamination reactions, the amino nitrogen from amino acids are ultimately concentrated into glutamate amino acid. This is because glutamate is the only amino acid that undergoes oxidative deamination at an appreciable rate. Now the oxidative deamination. Transamination reactions does not result in any net deamination. Therefore, oxidative deamination takes place. In this reaction, an amino acid is converted into the corresponding keto acid by the removal of the amine functional group as ammonia, which eventually goes into the urea cycle. Enzyme involved is glutamate dehydrogenase, and the reaction requires an oxidizing agent, NAD plus or NADP plus. Now the catabolism of amino acids carbon skeletons. Amino acids can be grouped into classes based on the metabolic products formed due to degradation of their carbon skeletons. These metabolic products are acetyl coenzyme A, acetoacetyl coenzyme A, pyruvate, alpha ketoglutarate, succinyl coenzyme A, fumarate, and oxaloacetate. These molecules are used to synthesize fatty acids or glucose or to generate energy. Amino acids, therefore, can be further classified into three categories: glucogenic, ketogenic, and glucogenic and ketogenic. Glucogenic amino acids are those which produce glucose precursors, that is pyruvate, or TCA cycle intermediates such as alpha ketoglutarate or oxaloacetate. which produce glucose via gluconeogenesis. All amino acids except lysine and leucine are partly glucogenic. Amino acids lysine and leucine are ketogenic and give rise only to acetyl coenzyme A or acetoacetyl coenzyme A. A small group of amino acids, that is, isoleucine, phenylalanine, threonine, tryptophan, and tyrosine, give rise to both glucose and fatty acid precursors and thus are characterized as being glucogenic as well as ketogenic.